Hello everyone, welcome to my YouTube channel. Today we'll discuss about the last major topic of this module, that is importance of special biomolecules. So far we discussed about biomolecules, that is carbohydrates, proteins, nucleic acids, lipids, all those things we have discussed. Then we went on discussing about the stem cells, all those things. So coming back to the biomolecules now. So some special biomolecules are there. They doesn't fall under the category of the carbohydrates, proteins, nucleic acids, all those things, but sometimes protein will act as enzymes like that. So but some of the special biomolecules are there, like enzymes, hormones and vitamins, whereas enzymes separate properties, classification, functions and all you have to explain, whereas hormones and vitamins slowly if you have given the smaller details also is fine according to your syllabus. So let's start with the enzymes. So enzymes are nothing but they are essential biomolecules. Definitely what they do, they catalyze the biochemical reaction in living organisms. That means enzymes are taken in different subjects in different way. Either they have to enhance some reaction or they have to reduce some reaction, they inhibit some reactions, all these things. But in biology most of the times these enzymes are going to do all these characters, but especially for biochemical reactions they will be enhancing or catalyzing some particular reactions. So those are called enzymes. So enzymes are nothing but what? Essential biomolecules. That means they are required. So for what? Catalyzing the biochemical reactions in living organisms. So they are very crucial for what? Maintaining metabolic pathways. Okay, I have discussed in the previous class in the same module. So facilitating the cellular processes. Okay, and also regulating biochemical reactions. Okay, these are the three major functions they do: maintains the metabolic pathway, facilitates the cellular processes and also regulates biochemical reactions. So enzymes play a very fundamental role in especially digestion. We are having trypsin, pepsin, all these kind of enzymes are there. When we eat food, it will go to stomach and those that food has to be broke down because it has to be absorbed in the intestine. So for that the food has to be digested. So for that we are having digestive enzymes which work at pH 1 to 2. Okay, imagine that pH, very acidic. Next, energy productions. During energy productions also in biochemical pathways, like for example we are having glycolysis pathway. In that NAP, NADPH, okay, when that conversion is happening in order to get your ATP as a source of energy, there are two kind of enzymes which will be play a very important role. Not only two kind but many will be there, but NADP, NADPH and all these enzymes plays a very important role in order to finish this biochemical reactions. Okay, so that you will get the energy, that is energy production. Then DNA replication. For DNA replication, what you have to do, you have to make a copies of DNA. For that what you need to have, okay, you will cut the DNA, right? Once you cut the DNA, what you want for cutting the DNA, you need restriction enzymes. That is also comes under enzyme. And for making number of copies, what you want, you have to give them more nucleotides. Okay, so nucleotides should be given to make more number of copies. So who is giving that nucleotides? It's Taq polymerase enzyme. So like that, so many enzymes will be there. Okay, they follow in digestion, they will be they are a part of energy production, they are a part of DNA replication. Without enzymes these processes are not gonna happen. That's why they are called as essential. Okay, and protein synthesis also we need that. Okay, and then numerous other physiological processes also we need that. So classification, we are having many enzymes, several groups are there. We can categorize into many groups. So some which are based on the functions we are going to categorize here. Oxidoreductase enzymes. So two reactions everybody knows: oxidation, reduction. Based on this, okay, we are having based on the electron transfer we are having these reactions. Either they are facilitating oxidation, either they are facilitating the reduction. Okay, so example we are having alcohol dehydrogenase. This is one of the enzyme. So what it will do, this converts ethanol into acetaldehyde. Okay, during the alcohol metabolism it converts ethanol into acetaldehyde. Okay, so that's how this enzyme play a very important role. So next, hydrolases. Hydrolases means what? hydrolysis reactions it has to catalyze. Why? Because what you mean by hydrolysis? Breakdown of the water molecules. So breaking down the molecules by adding water. Now example, so we are having lipase is one of the example of hydrolysis enzyme. So what this lipase will do, it hydrolyzes lipid, that means in the fat it will add the water molecule to break it into glycerol and fatty acids during digestion. Okay, so that's what lipase does. So we are having oxidative reductase enzyme, example is alcohol dehydrogenase. Hydrolysis, we are having example lipase. Next transferases. So this is especially when you are transferring the functional group. It can be amine group or phosphate group. Why we have to transfer this? Because many physiological processes cycles follows these reactions. So we are having phosphorylation, in that transferring phosphor group from one molecule to another molecule, like that. So catalyzed transfer of a functional groups like amine groups or phosphate groups between the two different molecules. Example, we are having kinases which transfer phosphate groups from ATP to specific substances. So we are having ATP 3-phosphate, adenosine triphosphates are there. Now one phosphate will be transferred to the specific substrates of an example proteins. Okay, that is what is happening in phosphorylation reactions. That is what I have given example. It's just you have to, non-biological students have to just think that we are just transferring a particular part of a molecule from one place to another place, from one molecule to another molecule. Okay, that's example is kinase, which is used in phosphorylation reaction. So which transfer the phosphate molecules or groups. Okay, just what you have to remember. Next, isomerases. So catalyzes the rearrangement of the atoms within the molecule. In the same molecule, there it is the entire group is transferred. Here isomerases, atoms, the molecule is same, only the atoms will be altered. Okay, so converting one isomer into another. So one isomer will become another isomer. Example, triose E-phosphate, triose phosphate, what we call isomerase we call. Okay, so this isomerase which catalyzes the interconversion of a dihydroxyacetone phosphate. This there is a conversion. Interconversion means the atoms in this particular molecule will be interchanged, and glyceraldehyde 3-phosphate during the glycolysis pathway. So we are having the 4 enzymes we already discussed, that is oxidative reductase, example is alcohol dehydrogenase, hydrolysis, lipase is the example, transferases, kinase is the example, isomerases, that is triose phosphate is the example. Next, 2 more examples we will discuss, that is ligases, means attaching one. So synthetases also they are called, that means if 2 DNA molecules are cut, now I want to join that. Okay, so then I will be using ligases. So it catalyzes joining of 2 molecules, so often coupled with the hydrolysis of ATP. So example, DNA ligase, which joins the DNA fragments by catalyzing formation of phosphodiester bond. Okay, it will give the it will form the phosphodiester bond, so that 2 DNA fragments will combine together during DNA replication and as well as repair. So ligase example is DNA ligase. Next we are having lyases. So lyases, what it will do, either it catalyzes 2 things, either addition or removal of the groups to form double bonds or break double bonds. If you want to want double bond, it will add. If you want single bond, it will break. Okay, so it's addition or a removal of a group to form double bond or to break double bonds without hydrolysis or oxidation reduction processes. So it also works as the other enzymes, but here without hydrolysis, like for example isomerase works like that. So but here without hydrolysis. Okay, so example we are having carbonic anhydrase, which catalyzes the reversible hydration of the carbon dioxide to bicarbonate ion. That means I can also, if I add, I can go for carbon dioxide to bicarbonate. If I remove, I can go from bicarbonate ion into the carbon dioxide. So it's like it should be a reversible. Most of the lysis enzymes will be working in a reversible way only, carbonic anhydrase. So 6 enzymes, 6 examples. Talking about moving to the properties of enzymes, so they are highly specific. Why enzymes are very important? Because they are highly specific to a particular substrate. Imagine there is a substrate, there is an enzyme. When they combine, okay, you will get the product. Okay, so and these substrate and enzymes are highly specific, not just chemically but also structurally. Okay, so if any photos are there, I would like to show. Okay, photos I have not put on model. Structure, you just take your one hand and another hand with the perpendicular to each other. So imagine one of your hand or a palm as enzyme, another as a substrate, so both will fit together, right? So like that, so enzymes and substrate will fit together, then only you will get the product, or else you will not get. Understanding, they are highly specific. Next, catalytic activity, definitely they accelerate biochemical reactions by lowering the activation energy which is required for the reaction. Imagine, for activation energy is 100, so much I need. Okay, so but if I use enzyme, it accelerated the biochemical reaction. Why? I don't need so much energy now. Okay, so like that to proceed, so without being consumed or permanently altered in the process. So definitely enzyme will not be consumed in any reaction, it will act as just a supporting character. Next, temperature and pH sensitivity. Definitely enzymes have optimal temperature and pH conditions under which they can function most efficiently. At a particular temperature, at a particular pH, their activity will be very high. So if changes in temperature and pH outside the optimal range, then definitely they undergo denaturation. That means they will denature means they will degrade, and it will affect their activity also. So this is also one of the property. Okay, that is they are highly sensitive to temperature and pH. Next, regulation. So enzyme activity is regulated by various mechanisms. For example, we are having allosteric regulation, competitive and non-competitive inhibitions, covalent modifications, enzyme induction and repression. This all comes under regulation. Based on the what function they have been used, the regulation will changes. Now moving on to the functions. Metabolism, definitely we spoke a lot of things about metabolism. For biochem, from glycolysis pathway to the phosphorylation pathway, we need enzymes. Without enzyme, there is no metabolism. So they play a central role in metabolism, facilitating biochemical transformation of nutrients into energy. So we will get from the glucose to the energy ATP because of enzymes. Building blocks for macromolecules, they also act as a building block for macromolecules and waste products also. Next, digestion. Without enzymes we won't digest. So trypsin, pepsin and many enzymes are there, they are responsible for digestion. Okay, so without our digestive system, okay, efficiency whatever it is there, it will catalyze that system or process of digestion. So breaking down complex molecules, example what we eat as carbohydrates, maybe protein, sometimes lipids, where oil and other fat things are involved, they will reduce that into, break down into smaller pieces, then it will make sure that they are absorbable molecules by the body in intestines. So next, speaking about DNA replication repair, we already discussed. If you want to replicate DNA, you have to cut the DNA. For that, restriction enzymes are required. Then you have to add something to it. Then for that, ligase enzyme is required. Then you have to keep on adding. If you want more number of copies of the DNA, you have to keep on adding nucleotides. For that, tag polymerase you need. Okay, for all these things you need to go for enzymes. Next, protease synthesis. Protein synthesis also requires enzyme. Okay, we are having RNA synthesis, one of the enzyme, and then for protein synthesis also you need. Okay, transcription means RNA synthesis, translation means protein synthesis. From DNA it will go under transcription, it will give mRNA, so that is RNA synthesis. From transcription it will go to translation to give the mRNA to the protein. For that also a series of enzymes are involved. Next, detoxification. So liver has to be detoxificated. Most of our bodies has to undergo detoxification. That means removal of the waste things which are present in that organ, especially in the liver. It has to be done effectively, or else if liver doesn't work, there is no digestion. Even though digestion happens, it is of no use, because all metabolism takes place in the liver. Okay, and definitely pancreas will be helping that. If anything goes wrong with the liver, then everything will collapse. So enzymes in the liver and other organs facilitate the metabolism and detoxification of the xenobiotics, that is foreign compounds, drugs, toxins, and making them more water soluble and easier to exit from the body. That's why you might have seen whenever you take some strong medicines, okay, or some normal medicines also, you will pass your whatever the urine you pass, it will be colored urine. Sometimes it will be greenish, sometimes yellowish, very deep color. Okay, why? Because our body has been detoxified that particular extra waste products or which are coming from the drugs. Okay, because the drugs will not carry only drug, along with that lot of waste product will be there, because it has to go through our system, our digestive system, all those things. So some extra added sugars or molecules will be there. So in summary, enzymes are very crucial or critical biomolecules that catalyze the biochemical reactions, and they regulate metabolic pathways, enables efficient functioning of the living organism. In short, we need enzymes for biochemical reactions, metabolic pathways, and also if you want effective functioning of your body, enzymes are must. So next, understanding their classification, properties, function is very essential for elucidating the complexity of biological system, or else if you don't understand their properties and functions or classification, you will end up with misunderstanding of complex biological systems, including the physiological pathways, biochemical pathways also. So developing a therapeutic and as well as intervention of the various diseases also will be difficult. So that's why understanding of enzymes plays a very important role. Next topic, we are having the hormones, that we will discuss in the next class.